Corn protein discovery boosts seed longevity and resilience. A new international study led by the University of Kentucky reveals a critical genetic factor that significantly improves corn seed viability during storage. The research identifies a specific protein repair L-isosparatyl methyltransferase 1, ZM-PIMT1, and demonstrates how variations in its regulation directly impact a seed's ability to withstand aging and harsh conditions. This discovery offers plant breeders a precise target for developing more robust and long-lasting corn varieties, reducing waste and improving agricultural efficiency. The critical role of ZM-PIMT1. The study, published in the Plant Cell, focuses on the enzyme ZM-PIMT1, which plays a vital role in repairing damaged proteins within seeds. Over time, natural chemical reactions degrade essential proteins needed for germination. Instead of complete protein replacement, an energy-intensive process ZM-PIMT1 flips faulty protein pieces back into working shape, conserving resources and ensuring critical systems remain operational during the initial stages of germination. Researchers found that natural genetic changes in the regulatory region controlling ZM-PIMT1 expression, the on-off switch for the gene, directly correlate with seed aging tolerance. Some corn lines possess a version of this region that strongly activates ZM-PIMT1 production, resulting in superior seed survival during long-term storage. Two versions, different outcomes. The research team, including collaborators at Northwest A and AMP University in China, identified two primary versions of the ZM-PIMT1 regulatory region. One version drives high ZM-PIMT1 mRNA production, leading to robust protein synthesis. The other carries a large DNA insertion that lowers expression, weakening seed performance under stress. Seeds with higher ZM-PIMT1 levels maintained higher germination percentages and produced healthier seedlings after accelerated aging tests, a standard measure of seed storability. Why this matters? Food security and economic impact. The discovery has far-reaching implications. Roughly 70% of the human diet relies directly on seeds, with the remainder dependent on animals fed on seed-based feed. Seed failure translates into substantial economic losses for farmers, seed companies, and consumers. Choosing corn lines with stronger ZM-PIMT1 expression is a practical step toward more reliable seed lots. Cellular repair in action. ZM-PIMT1 primary function is to repair a protein called PABP2, which helps select which stored messages get turned into new proteins when a seed germinates. If PABP2 is damaged and not fixed, the seed cannot make important proteins quickly, losing vigor. With higher ZM-PIMT1 levels, PABP2 functions better, allowing the seed to handle aging and still sprout strongly. Practical applications for breeders. The study reinforces the importance of protecting protein synthesis machinery for seed survival. By providing breeders with a concrete genetic marker to track, the research offers a targeted approach to developing more resilient corn varieties. Choosing lines with the stronger ZM-PIMT1 promoter is a practical step towards seed lots that stay reliable. The broader context. The discovery highlights the power of cross-disciplinary research, combining molecular biology with agricultural resilience. Understanding how seeds survive drying, resist damage, and complete germination is critical for food security and conservation. The research underscores the importance of basic science in addressing practical challenges in agriculture. In conclusion, this study provides a clear genetic target for improving corn seed longevity and resilience. By focusing on the ZM-PIMT1 protein and its regulatory region, breeders can develop more robust varieties, reducing waste and strengthening the global food supply.